10 degrees per second. The eye responded with a latency, so 100 milliseconds of nothing, a rapid change in eye velocity, and accurate tracking. And when the animal made this rapid saccade, the velocity was very fast, well into the basement of this building. Now, as I say, this is the evidence that pursuit responds to target motion and saccades respond to target position. It's part of our divide and conquer strategy. But the next part of that strategy is actually to get rid of the saccade altogether, because I know lots of people are interested in saccades, but they're in our way. We want to study pursuit. And so what we do is to simply take the step in target position and make it smaller so that now this displacement of the target exactly compensates for the latency of the monkey's response, and when the target catches up with the monkey, the eye has accelerated and he's tracking perfectly. Okay. In this world, the animal is tracking a target that's moving through space. But the visual input that drives that target is the target motion on the retina. And that's the difference between the motion of the target and the motion of the eye. And so here I filled in the difference between target and eye velocity, and this is how much the target is moving across the retina. So you can see out here that the animal is tracking beautifully, and so the target's not moving on the retina, and so this isn't even a visually driven part of the response. There must be something other than vision that's driving the response here. But here, we return to our spider lady ball girl, who took a glimpse of the target, which is what's happening here before the eye starts to move, and then generated this beautiful response, almost perfectly accurate, bringing the eye up to target velocity before there's been any time to process the visual inputs from here. And so now we have returned to the situation where we have a sensory stimulus and a motor response, and we can use this to ask how the brain generates the motor response. Okay. I've made the point here that the stimulus for the system is image motion, and so now we can think about the initiation of pursuit as involving converting some neural representation of image motion into commands for eye direction and speed. And so we have a sensory representation of image motion and a motor command in eye direction and speed, and that's what we think of the pursuit system doing. But remember, we're interested in how the pursuit system does it. So we need to dig deeper. I think of this as peeling the layers off an onion. And what I'm trying to do for you is peel the layers off the onion one at a time until we get closer to the core of the onion. Not all the way there, but we're getting there. One of the advantages of this system is that we know the neuroanatomy. The neuroanatomy is as follows. Visual inputs arise from the retina. They go through the lateral geniculate nucleus to the primary visual cortex, which is the area that Hubel and Wiesel studied. From the primary visual cortex, signals then are distributed to myriad other visual areas, one of which, the best studied and best understood of which, is called the middle temporal visual area, or if you're in Great Britain, V5, and to us affectionately known as MT for middle temporal. As I will show you shortly, neurons in MT respond selectively to motion, and they're tuned for the speed and direction of motion. Now, signals from MT then are distributed across the cortex into parietal areas and to motor cortex, and all of these go down to the brainstem and the cerebellum and out through the extraocular muscles to control the eyes. There are only 8 synapses, and as I will show you, we already know a lot about MT and a lot about the cerebellum, so what we really need to understand is how the signals in MT are converted into the signals in the cerebellum. Okay, let's try to take this problem now, which is getting from vision to eye motion, and move back into it. So let's see what we are looking for, what the motor activity looks like. And this is people don't show this kind of data. Oops, so I told you the hippocampus is a circuit. The cerebellum is also a circuit. It's highly structured, and this circuit is very intricately involved in motor control. And this is the sort of data, people don't show this very much anymore. This part is familiar to you, right? It's target motion, eye motion, Target velocity, eye velocity. It's the animal doing a pursuit trial. This is the Purkinje cell. It's a gorgeous cell, remarkably complex dendrite. This is the cell body and the axon going down, mostly going to deep cerebellar nuclei, but also giving some axons that go back to neighboring Purkinje cells. And these are recordings made by taking a microelectrode, running it down very close to the cell body of a Purkinje cell, and listening in on its electrical activity while the animal made this eye movement. And so these spikes are action potentials, and their frequency encodes information. These neurons emit action potentials constantly when the eyes aren't moving, and when the pursuit is initiated, they initiate a burst of action potentials during the rapid eye acceleration, and then a higher firing rate during the eye velocity that relaxes back to the baseline. So this is the motor command that we're trying to generate, and we're going to generate it from activity in area MT. And so I'm now going to show you how MT neurons respond. And what I'm going to do is I'm going to take recordings like the one I just showed you, but instead of showing you the spike trains, I'm going to play them through the audio monitor so you'll hear the spikes from the neuron. And this is the first movie. And listen in. And I forgot to mention that you're the monkey, but you're obviously doing your task. Your task is to fixate right here. And it's important for you to fixate there because I'm recording from an MT neuron that responds to stimuli presented in this part of the visual field. So if you fixate over here, you'll take the part of the retina that this neuron responds to off the cortex. Can you play that again? Okay, so this neuron responds only for targets moving to the left or left and up or left and down. It is tuned for direction. And the next movie is another neuron, but the same general idea. Different speeds. This neuron is tuned for speed. It prefers middle speeds. That's how neurons in MT respond. Okay, so the brain needs to convert visual motion into eye motion, but we can change that now. We can now say that the brain needs to convert the activity in area MT into cerebellar activity. To do that, it needs to estimate image speed and direction to tell the motor system about it. And this is embedded not in single neurons, but in the responses of the large population of neurons. So we need to understand how you take the response in MT and figure out what the target did. And you need to do this for perception too. And how? Well, we're going to divide and conquer. First, we're going to understand what the population response means. And second, we're going to figure out how the brain uses that population response to estimate image speed. Okay, how do we measure the population response? In neurophysiology, it is typical to run your electrode down into the brain and you isolate the electrical activity from a neuron and then you cheer because you know you're going to get data and your day is going to be successful. And when you get there, you then stay with that neuron and you record it with every stimulus you can think of. And so here, for example, are recordings from they're fake, but schematics of recordings from 3 neurons. And these are MT neurons. And these plot firing rate as a function of stimulus speed. So this is a neuron that prefers slow speeds, and it's tuned like the neuron I showed you. This is a neuron that prefers medium speeds and is tuned. And this is a neuron that prefers high speeds. Well, that's great, but the brain doesn't know about how these neurons respond to all the other stimuli. All it knows is that something moved and neurons responded. And so what the brain knows is a single train of action potentials from each of these neurons. So to assemble that, let's assume that we presented target motion at 16 degrees per second. And let's then take the response of the blue neuron and plot it as a function of its preferred speed, and the response of the red neuron and plot it as a function of its preferred speed, and the response of the green neuron and plot it as a function of its preferred speed. And now, if you were looking at this population response of only 3 neurons, you could tell me that the speed of the target was probably around 12 to 16 degrees per second. It's the start of a population response. Here's a real population response. Here, each dot represents the response of a single neuron recorded by my student Mark Churchland in MT. And if you look at this population response, where each neuron is plotted as a function of its preferred speed, you would say, well, the neurons that are responding the most are the ones that are tuned for 16 to 20 degrees per second. That must be how fast the target was moving. Okay, well, that's easy for us to do intuitively, but how's the brain do it? Remember, in this system, the brain has 100 milliseconds to do this. So it has to do it very quickly. This is definitely pre-conscious behavior, right? This has nothing to do with consciousness, nothing to do with thought, nothing to do with looking at the population response and thinking about it. It's something the brain has to compute with a circuit. So let's create this imaginary population, and let's record from it for target motion at 10 and 6 degrees per second. And this is what we get. Now, I'm going to give you one more piece of... Okay, so actually I'm ahead of myself. Can we figure out what the speed of the target motion was by simply adding up how many spikes there were? Well, the answer is no, because in both cases, the population gave off 18 spikes, but you can see that different neurons were responding. So we must need to know which neurons were responding. I'm now going to give you the additional piece of information that these neurons are tuned for different speeds. And so now you'd look at this response and you'd say, yep, 10 degrees per second. And you'd look at this one and say, yeah, 6 degrees per second. But how do you do that in 100 milliseconds? Well, you do it by computing what's known as the vector average. And the way you do that is you take each neuron, you take its preferred speed, and multiply by the number of spikes the cell emitted. And the same thing here, preferred speed of 10, 6 spikes. And you do that all the way through. These are the votes of each of these model neurons. You add them up, divide by the total amount of activity, and bingo, 10. Same thing for the 6 degree per second. These are the total number of spikes multiplied by the preferred speeds. Add them up, divide by the total number of spikes, 6. So now we have an equation. We've gone from saying you estimate the preferred speed of the neurons that have the biggest response in the population to saying this is an equation. If we could implement this equation in neurons, then we could do this in 100 milliseconds. So vector averaging reports the preferred speed of the most active neurons. But does the brain really do something like this? Well, how would you figure that out? Well, Mark Churchland figured it out. And his approach was to stumble on, and make no mistake, a lot of science is serendipitous. We set out to do this experiment for the completely different, very valid reasons. But Mark's a very observant young man. And he noticed something that was the following. He noticed that he noticed a situation where the population response in MT became smaller in magnitude, but the peak moved towards neurons that preferred higher speeds. And so if the pursuit system was estimating target speed by counting the number of spikes, it would say the speed of the target slower. And if it was estimating it by finding the peak of the population response, it would say the speed of the target was faster. And here's the stimulus he used. And can you start the movie? These are two different targets. This one is sampled motion or apparent motion, and this one is smooth motion, or as smooth as we ever see, right? We mostly see motion on computer screens nowadays. That's sampled at 100 hertz. This is sampled at a little lower rate. And then he looked at the eye movements evoked by this, and if you can start the movie again, this is sort of like going to the ballpark. Here's the animal's eye position, and lo and behold, the sampled motion caused a faster response, so that the eye position got ahead. And you can see in these eye velocity traces that for the sampled motion, a little delay, but then a much bigger estimate of target speed. Now, embed these in your visual memory, because they're going to appear on the next slide right here. Okay, you can forget them now because they're in front of you. Eye velocity is a function of time. Smooth motion, an estimate of faster speed. And smooth motion, estimate of lower speed, sampled motion, estimate of faster speed. Mark then recorded the population response in MT. Now, we've been through this. We've seen these kinds of graphs. These are the tuning curves of 5 different cartoon neurons, each preferring a different speed. And in the smooth motion, they all had the same peak response, but when the motion was apparent motion, the sampled motion, these neurons had smaller responses. So this is a situation where the